Thank you very much for attending this lecture about how artificial intelligence is enhancing human microbiome research. So I am a research scientist in artificial intelligence and data science for microbiome data. It's been 7 years that I work at Metagenopolis in France. It's a research unit specialized in the human gut microbiome. So first, in this lecture, I'm going to define some terms of the artificial intelligence or AI. And after, I will show you specific example of the use of artificial intelligence for microbiome data. And to finish, we will see the current artificial intelligence challenges for microbiome data. So first, let's define some terms, because artificial intelligence is a very trendy topic. As you may see on this figure, it's a number of publications containing the terms artificial intelligence or machine learning or deep learning, and it's really increasing in the last years. And if you add keywords like health or genetic or genomics, it's having the same trend. So I took this figure from the report for artificial intelligence for genomic medicine. But what do we mean by artificial intelligence? Artificial intelligence, according to this same report, it's the development and use of computing systems concerned with making machines work in an intelligent way. So as you can see, it's a very broad definition. So generally, artificial intelligence is divided in two categories. The first category is narrow artificial intelligence that focuses on performing a specific task, for example, play chess or filter spam or whatever. And although they are named narrow, they rely on sophisticated technology. So currently, narrow artificial intelligence is at the center of most current artificial intelligence development in healthcare and other industries. The second category is general artificial intelligence, which refers to a sentient machine with human reasoning. So I will break your dreams now. This artificial intelligence does not currently exist, and many consider that it's totally unachievable. So we are all going to talk today about this narrow artificial intelligence. So this is a quite broad field, and in this field, there is machine learning that we are going to talk about, and deep learning, which is a specific subcase of machine learning. So the first generation of artificial intelligence for medicine were a rule-based system. It was a system where you encode the knowledge of a human expert in an automated system. You have the input, for example, the symptoms of a person. You have different rules depending on, for example, different tests or examination. And depending on the results, you have the output. So the advantages of this system is that it was highly interpretable. For a given output, you know exactly what rules had been applied. But the limitation of such system is that it's expensive and long to build. You need a in-depth expertise of a human. It's difficult to develop when you have a complex system, and it's quite rigid because it can be updated only manually. So after this first system, the machine learning had been developed. So machine learning it's a set of methods based on algorithms that use mathematical procedure to analyze data structuring. So you provide a ton of data to the algorithm, and the algorithm will, depending on this data, take some decision. So the advantages is that it's less demanding to build because it's data driven. It's less difficult to encode, and it's more flexible to, for example, integrate new data. But it's more difficult to interpret, especially for deep learning, but we will discuss this a little bit later. So I put here the citation of artificial intelligence pioneer who says that machine learning is a field of study that gives computers the ability to learn without being explicitly programmed. So that was a huge step in artificial intelligence. So we are mainly going to focus on machine learning in this lecture, and we will review the three types of machine learning algorithms. So the first one is the unsupervised learning. When you need data that have no labels, you won't have any feedback from the algorithm. For example, you don't have an accuracy metric or things like that. And this type of algorithm will help to find an underlying structure in the data, for example, to find clusters of data. The second type is supervised learning. To do supervised learning, you need labeled data. You need to know, for example, who is healthy, who is diseased. And this algorithm will give you a direct feedback with a performance metric or accuracy metric to know if the performance of the algorithm is good or not. And this kind of algorithm will provide the prediction of an output for new data. For example, if you have a new individual, is it a healthy individual or diseased individual? And finally, there is this third category of reinforcement learning, where you do not need labels, but you have to provide a set of rules. And it's probably the closest procedure to how we humans learn. The algorithm learns from its environment by interacting with it. It gets a positive or negative reward based on its action, and so it's a self-teaching iterative. So reinforcement learning is behind self-driving cars and the famous AlphaGo programs that won over humans at the Go game. So let's see how unsupervised learning works. The first step, you need your data. So here, data are picture of dogs or cats. And you can choose algorithm, the popular algorithm to do unsupervised learning. There are K-means, hierarchical clustering, or principal component analysis. So those kind of algorithm. And it will provide you groups of data that are similar together. And hopefully this structure will help you to make sense out of your data. So here it's a good example because algorithm has divided the cats together and has the clusters dogs together. So the type of problem you can answer with unsupervised learning is clustering to identify similarities in groups, anomaly detection to identify abnormalities in the data, or dimensionality reduction in order to provide concise input for supervised learning with less reduced number of variables. Here you have the famous example of the fish. The fish here on this picture is in 3 dimensions. And you see that you can see it in 2 dimensions from face or from side. But if you look at this way from face, then you have the majority of information without losing much information. So that's the point of dimensionality reduction. You have less dimension and you try to keep somehow the majority of the information. So let's see now how we build a supervised learning model. So to build this, we first need to gather, select, prepare the relevant data that will be used to train the mathematical model of machine learning. The data preparation can include the cleaning and organizing data. That's often an important step, especially when we work with clinical data that always may have some problems in it. After, the second step is to select the learning approach. So there is really many approaches that exist. I just put 3 here. For example, lasso regression, random forest, support vector machine. So whatever a learning approach here can be chosen. And the third step here is to use the machine learning model with the training data set provided here. And improve this model with parameter optimization. And this process is performed iteratively until the model is well trained. And once the model gives good performances, this model can be deployed to be used on the new data. And there, if it's for a clinical application, it needs to meet some, for example, performance criteria or ethical and regulatory requirements, depending on the context of use. And so what about deep learning? Because a lot of people talk about deep learning. So I'm going to explain how it works. So deep learning, it rely on artificial neural network. So what is a neural network? It's a collection of connected units whose functioning is inspired by a neuron in the brain. So here you have a neuron in the brain with the cell body and dendrites and the axon here and the synapse. And so how a neuron is working, very briefly, some information, some chemical information reach the dendrites. And depending on the quantity of chemical component here, the axon will or not be activated. And some chemical component will be released here at the synapse. So here is an artificial neuron where you give some information in input. You have here some mathematical function, and depending on this mathematical function and of the input, it will or not provide some output here. And so the deep learning is a learning process based on the large artificial neural network with many hidden layers. So you can imagine here a network with many neurons, and here you have the chemical components that convey the information between the neurons. And here is artificial pendant of this, where you provide the data in entrance. You have the input layer with a few neurons, and then several hidden layers that will convey the information. And for each node here, for each neuron, you have weights that you can train based on the input data. And all this complex structure will provide an output, and hopefully the output will be close to what you are expected depending on what you have in entrance. So here, what you have to understand is that each hidden layer in the deep learning network detects and integrates information from patterns into neuron from the layer below. So in deep neural networks, you may have more than 100 layers that enable them to model very complex relationship between input and output. So this is what we can do with deep learning. Some very popular example, I took this picture from the Stanford University website. The deep learning here can see a picture and recognize its elements. For example, here it recognizes it's a woman in a white dress standing with a tennis racket, and each time it's pointing towards the element that has been recognized. 2 people in green behind her. So when you see this, you see that it's a really powerful algorithm, and it's completely astonished that some algorithm can have such a good understanding of such a complex picture. And that's why the main application of deep learning algorithms are for image recognition, for example, facial recognition and object detection, and also for natural language processing. For example, all the algorithms that will detect the speech of a person and propose a direct translation or stuff like that. So the advantages of such method is that it's very flexible to model a complex relationship, and it's not that much dependent on prior knowledge on the field. For example, for the picture modeling, you do not need to provide other information than the picture itself. But some limitation exists, obviously. It requires a huge amount of data. If you do not have enough data, it may do overfitting. Overfitting is when the model is too well trained to some specific aspect of the training data, and so it makes a bad generalization to other data. It's really costly in terms of computing requirements, and it may be difficult to interpret and to understand why this algorithm has given this precise output. So I'm going to illustrate this on some very classical example. It's about an algorithm, a deep learning algorithm, who classifies dog as dog or wolf from pictures. The algorithm was very accurate, but for some dogs, the algorithm was wrong, and especially the husky were often classified as wolf. And so people wanted to know why the algorithm takes the decision that the husky is wolf. And so what they did is ask the algorithm, which pixel did you use in your process to take this decision? And this is a surprising result that gave this experiment, because the pixel used to take the decision, it's actually background pixel. The algorithm did not take into account the face of the animal. It just took into account the white pixel, because white is snow, and in the training data set, usually wolves were on the snow background. So this algorithm did make the classification by looking if there is snow, then it's a wolf, otherwise it's a dog. So that's obviously not really what you expect, and it's to make you understand that even if the recognition is very astonishing, the algorithm do not understand what is a woman, what is a tennis racket. Okay, it's only sometimes very encoded in a very surprisingly way. So I took here to illustrate this default citation that says, the question of whether a computer can think is no more interesting than the question whether a submarine can swim. But even with this limitation, deep learning performances remains tremendous. So what about the application for microbiome? So before going much more in detail, I want to highlight some differences between microbiome data and other data on which deep learning algorithms work well. First of one is the nature of data. Images are a well-known modeling, because we know that there are pixels that have some colors, and pixels that are close one from each other often refer to the same object and stuff like that. But microbiome data are not so deeply understood. The second point is a dataset dimension, where for images, for example, we can have access to very large data set. For the training, the recognition of picture, the ImageNet database have more than 40 million of images. Obviously, we do not have 40 million of human microbiome yet. And deep learning algorithm can also rely on transfer learning, where it's possible to train a neural network on one image category and transfer it to another image. For example, you train a neural network to recognize a dog, and after you have just to change the 3 or 2 last layer to use the same algorithm and recognize a cat instead of a dog. So with microbiome data, we have much less data available and higher heterogeneity. And the last difference is the nature of the question. For image recognition, humans can solve the problem. If I show you a picture, you will be able to tell me what is on the picture. And here for microbiome data, human cannot solve the problem. If I show you the microbiome of an individual, you won't be able to tell me from this if he's healthy or he's diseased. But once this said, let's see some application of artificial intelligence for microbiome data. So I took back the 3 main types of machine learning algorithm, and we will see some unsupervised learning algorithm applied to microbiome data. So unsupervised learning methods are used for the identification of community types, also called enterotypes, that is used to stratify individuals into specific subgroups based on their microbiome composition. Here, for example, you have a principal coordinate analysis that indicates sample similarity based on species level taxonomic composition. So here you have individuals that are dominated by Prevotella, here individuals that are dominated by Bacteroides and stuff like that. So when you have such a stratification, you may wonder where are you located depending on the other individuals. So different methods exist for dividing up the landscape of possible microbiome configuration. And the interest of doing this is to have a link with the functional, ecological, medical information. However, there is some controversy about this method because there is a lot of different ways to calculate distances and to perform some normalization on the data. And so some people say that it's hard to have really meaningful clusters. And here is an example of some clusters that can be strong. We can think that they are strong just because of the color. For example, you have some data points here in this figure, and here you have the color with 3 clusters and 3 different colors. And if we remove all color, that are the data points. And as you can see, it's much more difficult to see the structure in the data. And we can really wonder, does this structure in 3 clusters really exist here? So the second method that is used in unsupervised learning is network inference to infer a network of bacterial species. It's a quite popular explorative method of data analysis. Here you have a very nice paper about open challenges for microbial network construction and analysis. Here on this figure, you have the reconstruction of a network for liver cirrhosis cohort. So as you can see, a lot of species are not connected together, but you have 2 main gills, one who is green and one who is orange. You can analyze this to identify the taxa that may share a common role in the ecosystem. For example, here you have the all this guild zoomed here, and it's all taxa that are linked with the disease. And it's also possible to have a zoom on some specific bacteria and see the close network of this bacteria with others. So this is a very useful way to find patterns in microbiome data. And finally, the third way of using unsupervised learning algorithm is this dimensionality reduction I told you about a little bit before. If you are working on shotgun data, then you have the counting at gene level, and the catalog of gene for a human currently have 10 million genes. So it's a lot of variables, and it's very complicated to make relevant statistical analysis with 10 million variables. So if you cluster gene together, you can have this MGS for metagenomic species level, which is a proxy for bacteria, and which is much more relevant in biological, from a biological point of view. Here in this publication, you have some explanation on how to perform this metagenomic species reconstruction if you are interested. And the same way, from this bacteria, we can make network inference and find guilds of co-abundant bacteria, and so there is much less guilds. So we can, from a same data set, come from 10 million variables to thousands of variables to tens of variables. And hopefully we hope to have more biological relevant variables when we cluster information together. So let's see now what we can do with supervised learning applied to microbiome data. So for this, you need label data. So you need some microbiome data and information, for example, who is healthy and who is diseased. The common algorithm used for the disease prediction task, there are random forest, support vector machine, gradient boosting, lasso ridge or elastic net regression, partial least square neural network, etc. You have a very nice review here on the different algorithms that have been used in the literature. And some popular machine learning tools to perform this disease prediction task are basically R and Python and Weka, and in R the Carrot framework or MLR or tidy models. And I put here an example, which is quite a reference in the domain. It's a publication from Eduardo Pasoli in 2016, which is called Machine Learning Meta-Analysis of Large Metagenomic Dataset. So he took different data set of different diseases. So 6 different diseases, liver cirrhosis, colorectal cancer, IBD, obesity, and type 2 diabetes with 2 different cohorts. And what you can see here, it's a result of the disease prediction task. So here on the dotted line, you have the shuffled labels. Shuffled labels means that there is nothing to predict because you have shuffled the information on who is diseased and who is healthy. So that's the baseline. If the algorithm is not doing better than this, it means that it's not working at all. And what you can see is that all the algorithms are better than this baseline, but there is huge differences between the diseases. For example, for liver cirrhosis or even IBD or colorectal cancer, the prediction accuracy is quite good, and the algorithm can distinguish diseased and healthy people quite well. But for some other diseases, for example, for obesity, it's quite hard to make a precise answer. So there is still much room for improvement of such algorithm, but this paper and many other show that machine learning works on microbiome data, and there is a lot of differences between the different data sets. So let's talk now about what you are all waiting, deep learning. What can we do with deep learning on microbiome data? So I told you before that deep learning are well for image recognition, and it's true, but it was a little bit simplified because there is different architecture types. So the types of architecture that are well suited for image-based recognition are called convolutional neural networks, CNN. And so you provide the image, the algorithm will analyze the different localization of pixels and construct some layers to give the final answer of what is on the picture. The problem is that in microbiome, we do not have images. We have abundances of species for different individuals. So some people had the idea to convert this abundance information into image in order to use after CNN algorithm to make image recognition. But it did not work that well, and it's perfectly understandable because when you are converting microbiome data into image, then you are losing information, because when you are doing this conversion, you do not have 100% of your information. So it was clever to try, but it did not give really outstanding results. After you have the deep neural network, who have many hidden layers. But for some authors think that it's impossible to use for microbiome data because there is not enough data, because you need really a lot of data to train accurate DNNs. There are the recurrent neural networks, which are well used in speech recognition, that take into account the fact that elements are linked together and there is some temporality between the first and the last elements. So there is nearly nobody using this recurrent neural network for microbiome data, so I won't talk much about it. And the last part is about the last architecture type is about autoencoders. So autoencoders is a way to encode the information in some latent representation of reduced size. And the first step is to encode and decode the information and trying to have the similar output, the output the closest to the input. And after using this way of encoding information to have a latent representation and to make a dimensionality reduction, like I showed you before, we can do with, for example, PCA. So this has been used in some publication from MinO that was published last year. And what is interesting is that they used the same six data sets that were used in the Pasoli publication. So the Pasoli publication is considered as a reference, and here it's in green. So they made a dimensionality reduction based on autoencoder deep learning here, called the deep micro in red. And they compared also with two other ways to do dimensionality reduction, so classical principal component analysis, and some other way to do dimension reduction, who is called RP for random projection. So what you can see on this figure, you have the AUC here, and you see that the results are a little bit disappointing. because, for example, for IBD, the deep micro methods did not perform well than the original classical machine learning. For type 2 diabetes, it's a little bit better, but obviously it's not, the difference is not significant. And there is only, you see, for obesity, which is quite hard to predict, that deep learning based on autoencoder performs a little bit better than the original method. But for other, there was not so very exceptional results. So there is a very nice review of Nathan Lapierre published 2 years ago on this deep learning methods applied to microbiome data. And in the discussion, he says something important, that to say that considerable effort has gone into increasingly powerful deep learning algorithm, but with only minor improvement in the performance and modest changes in the ranking of the importance of feature. So all this to say that it's quite hard to have the same great result with microbiome data that the deep learning could reach in other domains. And finally, the last part is about supervised learning. Sorry, the reinforcement learning. So I first thought that there was not much people using reinforcement learning in microbiome data, but after some search, I found some publications that use the generative adversarial network for this. So it's a preprint. It's not yet published, but I find the way of doing quite interesting. They did use the reinforcement learning algorithm not to make the prediction, but to generate new data, to generate simulated data that really look close to the original data. So it's what you can see here illustrates a data-driven simulation of microbiome using the generative adversarial network. Here you have the original data with the IBD individual in red and healthy people in blue. And here you have the cross, it's simulated data, same for IBD and for healthy. And here you have the combined between original and healthy, between original and simulated. And only from a visual perspective, it seems that it's quite close. You cannot really distinguish the simulated from the original data. So it seems quite a good simulation. And so they showed here that a machine learning algorithm only on original data or on the original data plus simulated data gives better performances to make the prediction. So they increased the performance of the prediction by adding simulated data that were generated with a generative adversarial network. So they conclude that synthetic samples can boost disease prediction, and I found that it was quite an interesting result. So we are done with this application part, and we are now going to discuss some challenges in artificial intelligence for microbiome data. So the first challenge is obviously the misuse of a machine learning model. I find here a systematic review of human gut microbiome research. And this paper, which is a preprint, suggests that there is a widespread misuse of machine learning. And for this, the author says that a lot of publications do not integrate the good step to verify that the algorithm gives a trusted answer, AUC. For example, in some publication, authors do not use cross-validation to validate their models. And you can say that the reported AUC is higher for publication without cross-validation than the publication with cross-validation. And some papers also do not use external data set to validate the findings. And same here, the AUC reported is much higher when there is not a proper validation. And in this review of more than 100 articles, the author finds that 88% of the published AUC cannot be trusted. And he concludes that this finding casts serious doubt on the general validity of the research claiming that the gut microbiome has a high diagnostic or prognostic potential in human disease. So I find this conclusion is quite harsh, because there is still some serious publication using machine learning that shows that there is a diagnostic or prognostic power with gut microbiome data. But I think this is a very clear warning that with machine learning algorithm will always provide you some answer. And if you are not using the correct validation scheme, the conclusion can be totally wrong. The second challenge we have in microbiome data is the high inter-individual variability and currently limited data availability. I just take this example of high gene count and low gene count stratification population. This gene count richness is a good measure to distinguish, for example, healthy people from diseased people. Here you have this figure in the Queen et al. Paper about liver cirrhosis. And you see that diseased people have much less genes than the healthy people. And this is true in the discovery cohort and also in the validation cohort. And the p-value is very significant. But as you can see, there is still some healthy individual with really low gene count, and you still have a diseased individual with a quite high gene count. So even with something so significant, you have a huge variability between individuals. And I took this example, but it's for many other variables that you do not have a clear separation between category, and so it makes the learning task so difficult. So one thing that can help with this intervariability problem is having more samples. For that, Million Microbiome from Human project have been launched a few months ago. So it's a project that will analyze 1 million microbial sample from intestine, mouth, skin, reproductive tract, in order to build the world's largest database of human microbiome. So I think this initiative will provide a lot of data and will really enhance the possibility for machine learning and even deep learning, because with 1 million samples, it will become easier to make something without too much overfitting. So in this project, the founding members are from Europe and China, Sweden, Latvia, Denmark, and France. And I belong to the unit that is going to gather 100,000 samples for this million microbiome project. Another challenge for making artificial intelligence for microbiome data is a learning from multiple data sets. currently, the general approach is to gather different data sets from different places, different hospital, and to gather everything at a single place, and to perform machine learning or deep learning algorithm on it, and provide a model. But for a lot of reasons, this approach is not optimal. It's not optimal because patient data are usually sensitive data, and the hospital do not want to share or do not have the right to share the data. And putting all sensitive data in one place, it's quite a bad practice if there is some attack to this place, for example. Especially with now regulation like GPDR or things like that, this gathering a lot of medical information becomes less and less easy. And so one really active research area is federated learning approach, where instead of gathering all data at only one place, the federated machine learning approaches propose to share only the parameters of the models. So the data stay at their original location, and only the parameters of the model are shared and optimized in an iterative way in order to build this machine learning model without sharing any data between the different places. And finally, the last current challenge in machine learning is learning from heterogeneous data set. Because when we talk about microbiome, I talk a lot about metagenomic, but we can have also metatranscriptomic data, metaproteomic data, or metabolomic data. Plus additional clinical data, and for example, other type information like networks or images or something else. And the branch of artificial intelligence trying to integrate all this data is called multiview learning, and it's quite a hot topic currently to try to make sense of so much data concerning only a few number of samples. So my talk is nearly over. I will give you some message for home. We have seen today that machine learning is an important component of the growing field of artificial intelligence, and that deep learning is a subtype of machine learning with that is promising on some topics, and some people try to develop it for microbiome data. We have seen that unsupervised, supervised, and reinforcement learning are the three types of machine learning, and that they all have successfully been applied to microbiome data. And we have reviewed the current challenges and active research area that are the misuse of machine learning models, the high inter-individual variability, the federated learning, and the data integration. So I hope this lecture was helpful for you. I thank you for your attention. I thank you obviously the Cost Action and the Machine Learning for Microbiome group, and especially Dominika and Eliana for the organization of today.